Billahi minash shaitani rajim. Bismillahir rahmanir rahim. Assalamu alaikum, dear students. In the previous lecture, we have discussed the first part of the development of anther, that is development of anther wall, structure and development of anther wall. Now we will take it a second part, that is the most important part, that is sporogenous tissue. As we have seen, the archsuporial initials, they divide pericalinally to produce an outer primary parietal cell or layer and inner primary sporogenous cell. The outer primary parietal cell or layer, it divides by successive anticlinal and periclinal divisions to produce the wall of anther, to form the wall of the anther, which we have discussed in the previous lecture. And the inner primary sporogenous cell or layer, it divides further by various planes to form, by mitosis, it divides further in various planes by mitotic divisions to produce a number of microspore mother cells. Microspore mother cells or pollen mother cells. This group of microspore mother cells or pollen mother cells we call as sporogenous tissue. Sporogenous tissue. And in these cells, the inner sporogenous cells, they divide by mitotic divisions to produce a mass of cells or a tissue of cells. This tissue of cells or this mass of cells is called sporogenous tissue. It is a mass of microspore mother cells, and the cells which will produce microspores in the future are pollen mother cells, that is pollen grains. The cells which produce pollen grains. These pollen mother cells, and we get now large number of pollen mother cells. These pollen mother cells are connected by plasmodesmata. The different pollen mother cells which are produced here, they are connected with each other by special connections, what we call as plasmodesmata. These are connected with each other by plasmodesmatal connections. These plasmodesmatal connections finally break. These plasmodesmatal connections finally break and separate the pollen mother cells from each other. And each of these pollen cells, they are separated from each other, and separate these pollen mother cells or microspore mother cells. And in the early stage of the development, these pollen mother cells, they are connected by plasmodesmata with each other, which finally break and separate the pollen mother cells from each other. Some of these pollen mother cells degenerate. Some of these pollen mother cells degenerate and become nutrition and provide nutrition to the developing sporogenous cells. To the developing sporogenous cells or to the developing microspore mother cells or pollen mother cells, while as some cells, they act as pollen mother cells or microspore mother cells. And out of the large number of cells which are produced by the mitotic divisions of these inner sporogenous cells, some cells degenerate to provide the nutrition to the developing microspore mother cells, and only some cells, they take part in the formation of microspores, that is, they act as microspore mother cells or pollen mother cells. This is the sporogenous tissue. Now we will take up another aspect of this, that is microsporogenesis. Microsporogenesis. Now we have seen that some these microspore mother cells, they act as microspore mother cells, or some of the cells of the sporogenous tissue, they act as microspore mother cells or pollen mother cells, while other degenerate. These microspore mother cells, they undergo meiosis to produce microspores or pollen grains. With these microspore mother cells, they are diploid. They undergo meiosis to produce microspores or pollen grains. To produce microspores or pollen grains. This process of formation of microspores or pollen grains from the microspore mother cells or pollen mother cell, PMCs or MMCs is called microsporogenesis, and this process is called microsporogenesis. The microspore mother cells undergo meiosis to form a tetrad of, each of the characteristic feature of the meiosis is that one daughter cell it divides meiotically to produce 4 microspores which are arranged in tetrads. We get a tetrad of microspores. Tetrad of microspores of pollen grains. And the arrangement of these microspores, the arrangement of these microspores in a tetrad, and we now get a tetrad. The arrangement of these microspores in a tetrad depends upon cytokinesis during meiosis. And in the arrangement of these microspores, it depends upon the cytokinesis during meiosis. We see that the cytokinesis, that is the division of the cytoplasm, one is the division of the nucleus, that is karyokinesis. Second is the division of the cytokinesis. Karyokinesis results in the formation of 4 microspores, 4 nuclei from single nucleus. But these after karyokinesis, cytokinesis takes place. Now the arrangement of these microspores in a tetrad, it is affected by my cytokinesis during meiosis. It depends upon the cytokinesis during meiosis. And this cytokinesis, it is of 2 types. It may be of simultaneous type or successive type. And in the cytokinesis, it is of 2 types, successive type or simultaneous type. Successive type means when first and the second division of the meiosis, meiosis involves 2 divisions, meiosis first and meiosis second, both are followed by wall formation. That means after meiosis first we get first dyad, and then after meiosis second we get a tetrad. But in simultaneous type, both the cytokinesis, both the cytoplasmic divisions, they take place simultaneously. That means first meiotic division, meiosis first, it is not followed by wall formation. It results in the formation of a binucleate cell. And when this binucleate cell divides further, it produces a tetranucleate cell. And after the formation of 4 nuclei, cytokinesis takes place. That is, cytokinesis takes place simultaneously. It takes place only once, only after the completion of the meiosis, only after meiosis second. This type of this cytokinesis is called a simultaneous type. And the shape of the tetrad, it depends on this cytokinesis, whether it is simultaneous or successive type. Now the tetrads may be tetrahedral, isobilateral, linear, decussate, or T-shaped. And we get 5 types of tetrads. It may be tetrahedral, it may be tetrahedral, it may be linear, it may be decussate, that is cross-shaped, it may be T-shaped, even it may be T-shaped tetrad. And the shape of the tetrad, it depends upon the cytokinesis. And the microspores of a tetrad, they generally separate from each other. And after this meiosis sac, when a tetrad is formed, the microspores of a tetrad, the 4 microspores of a tetrad, they get separated from each other. They get separated from each other and form individual microspores or pollen grains. However, in Datura and Typha, all the 4 microspores, all the 4 microspores of a tetrad do not separate and thus form a compound pollen grain. Compound pollen grain. And we can say that in Datura and Typha, the 4 microspores of a tetrad, they do not separate, but they form a compound pollen grain. They found a compound, and a compound pollen grain is the characteristic of Datura and Typha. Similarly, in calotropes and some orchids, all the pollen grains of an anther, they remain united to form a typical structure called pollinium. And all the pollen grains, large number of pollen grains, large number of microspores are found, tetrads are found in a pollen sac. All the pollen grains of an anther lobe. Anther has 2 lobes, each lobe has 2 pollen sacs, and all the pollen grains of an anther lobe, they remain united to form a typical structure. That typical structure is called pollinium. That means pollinium is the characteristic of calotropes and some orchids. Sometimes question is asked, pollinium is the characteristic of Bankanchas, Typha, Datura, calotropes, orchids, some other kya there, and you have to remember that it is the characteristic of calotropes and orchids. Similarly, compound pollen grain is the characteristic of Datura and Typha. In all other these plants, generally pollen grains separate from each other, microspores separate from each other, but only in some, as in Datura and Typha, all the pollen grains of a tetrad, they remain united to form a compound pollen grain, and in case of calotropes and some orchids, all the pollen grains of an anther lobe, they remain united to form a typical structure that is called pollinium. This is all for today. Thank you. Assalamu alaikum.